Hi there, I'm Liz Bocardi,
I'm Carolyn Schultz.
and our paper is on behavioral changes induced by toxoplasma infections of rodents that are highly specific to aversion of cat odors by Voss et al. Information you need to know about the study is the nervous system, first of all. The brain and the spinal cord are main parts of it, but the brain is the focus point right here in the study. In the brain, there are neurons that carry signals from external stimuli to other parts of the individual, resulting in different behavior. So you have sensory neurons, which carry signals from a receptor organ to the central nervous system, and you have motor neurons, which carry signals away from the central nervous system, and they go straight to the muscles and the glands. So what do? Neurons are responsible for the way the individual's body reacts to certain stimuli, external or internal. Certain neural circuits are involved in the fear anxiety response to animals. Because of these neural circuits, they are innate. It's possible for the fear and anxiety responses to be altered by biological stimuli such as parasites. Rats were used in this experiment. So what we have here is the rats are innate aversion to predatory cat odors, and evolutionary and adaptation because this increases fitness and decreases predation. So the parasite attacks the body of the host, which is a protozoan parasite, Toxoplasma gondii, reproduces in the gut of the cats, but can also infect the rats. So the parasite causes cysts in the brain that are in the host, which cause the infection. So within this, there is a behavioral manipulation hypothesis. They call it a hypothesis in this study, but within broader context, and other further researchers have called this the behavioral manipulation theory. You will see us alternate, use them, go back and forth. So with this, many account for the abnormal behavior seen as the infected host of various parasites, and it's a really new concept. If you look here, we have included the life cycle of the toxoplasma. So first, it starts as an oovocyte, as you see right down here. And then you can see you can either go to a rat or a bird, and this then goes right into the tissue sites, as you see here, step 3. And then you can see our lovely cat here, who then gets it. And you can see it can either stay within the small red circle here, or it can go over to the left side, which is here, where it does start to affect humans. Now you may wonder, how does it get to humans? Well, it can get to us by, as you see, that either lamb or pig, if the meat isn't thoroughly washed or cut, or the animal could be infected, and if it's not properly cooked, then you can get infected that way. Another way, as you see with the produce there, that if the produce isn't washed correctly, that's another way of getting this toxic parasite inside of you.
For this paper, there's a lot of definitions that you need to be familiar with for your best understanding of it. So to begin, the behavioral manipulation theory or hypothesis that they talk about claims that a parasite can change the behavior of its host so that the parasite benefits from the host. So protozoan means that the parasite is a single-cell microscopic organism of the kingdom Protista. In vivo is the term used for anything biologically internal to the individual discussed. And so in the study, it was used for the bioluminescent imaging to track the bioluminescent signals from the parasites in the rats after being exposed to the infection. The photon flux was the spread of the bioluminescent light signals given off by the toxoplasma. Habituation is becoming used to or familiar with something over a period of time. This was used for several of the experiments in the study. Fibroblasts are cells and tissues that produce various fibers and collagen. This was where the toxoplasma was cultured for the experiment. Syringe lysis is just the technique of using a syringe to deposit the lyse cells. This is how they got the parasites into the rats and mice. Tachyzoids are the stage at which the toxoplasma are rapidly multiplying, and this is the stage where they injected the toxoplasma into the subjects. The phosphorus buffered saline is a water-based salt solution, and this was used as a placebo in the experiment. Luciferin is the organic compound responsible for the bioluminescence in the toxoplasma, and that's how they were able to use the bioluminescent imaging. The hypothesis of the study, they looked at how the behavioral manipulation theory conflicts with the current understandings of how parasites affect the behaviors in rats, aside from those just for the parasite's benefit. So there must be an explanation and distinction between both ideas from the previous literature's findings on toxoplasma in rats. The behavioral manipulation includes the theory that parasites can alter the behavior, but in the case of the toxoplasma, they reproduce in the stomach of cats, so the aversion to cat urine is the opposite of what the toxoplasma actually want from living inside the rats. So do they change the neuronal circuits to reduce the fear and anxiety rats feel when they pick up a scent of cat? That's what they're testing. Overall, the purpose of the study is to understand the behaviors of rats and mice after being infected with the Toxoplasma parasites. So they were looking at, do the parasites directly and specifically alter the behavior or behaviors for their own benefit, or do they just affect the host's brain and in turn cause non-specific changes?
Methodology of this experiment, they used male Long Evans rats, which are pictured in the bottom left, and female BALB/C mice, which are pictured in the top right. These subjects were found not to have toxoplasma prior to them at all. So what happened with these guys was that the infections were cultured in fibroblasts and syringe lysed into by using a 27 gauge needle to deliver the tegocytes, as mentioned earlier, which is the rapid forming version of this parasite in the cat's stomach. So the experimental conditions were subjects that were infected with the tegocytes. There were 106 for rats and 400 of the mice. And the control was their mock infection with the sterile PBS, which is phosphorus buffering saline. And there was a bioluminescent imaging that was used to see the movement of the infection of the toxoplasma in the rats and mice over time.
Continuing with the methodology, there were overall 6 experiments done. The first experiment they did was just on rats, and it was the open field exploration experiment. A ceramic plate was kept in their home cage for at least 2 weeks, placed in one quadrant, and it served as their home base. So they considered this their safe and familiar area in the arena for testing. So this test was used to identify and condition a safe space for the next test, the aversion to predator test. So in the aversion to predators test, they tested both mice and rats. The safe ceramic plate was put in one quadrant of the arena. In the other 2 quadrants, one plate had 20 undiluted drops of bobcat urine, and another plate had 20 undiluted drops of rabbit urine. The 4th quadrant had nothing in it. So this test was used to identify if the infected rodents were less, more, or the same adverse to different predators. The 3rd test they did was an aversion to the cat collar in mice. So in a rectangular plexiglass box, they placed a cat collar and an unscented sham collar in opposite sides. There was a habituation period before the stimuli were presented, but this test used to look specifically at the mice's reaction to predatory odors after infection. What they did looked at fear conditioning in just the rats. So they presented an auditory tones and a following foot shock on the rats. So the rats were conditioned to react to just the auditory tones. And over the next day, they measured the loss of the conditioning over 30 trials, and they correlated the conditioning to the infection. So this test was used to identify if the toxoplasma infection affected learned fear in rats, and this could be applied to how they affect the neuronal circuits in rodents' brains. Another test they did was neophobia towards food and food preference in mice. So mice were habituated to eat powdered food and subsequently food deprived for 12 hours. The mice were then presented with a scented and an unscented powdered food options. They measured the neophobia towards the unfamiliar scent. After they witnessed a demonstrator conspecific eat specific scented food, the scientists measured consumption of which food the test mouse ate after observing a demonstrator. They correlated this neophobia to the infection, and they used it to see if the infection really affected how afraid of new things the rodents would be. The experiment they looked at was spatial learning in rats. So they trained rats in a circular water maze to find a platform under the water. They recorded the latency during the 60-second trials, and they correlated the retention of this training to the infection. And they used this to see how spatial awareness was, if at all, affected by the parasitic infection. So these 6 different assays were used to identify the overall behavioral effects of the toxoplasma infection. Fear conditioning, neophobia, and spatial learning tests aim to rule out the confounding factors in the rodents. They also looked at cyst density. So after all the tests were completed and an ethical euthanation of the subjects, brain cysts were collected and used to identify the regions that they grew in and the overall density and size of them. So many tests were done in this experiment, there were a lot of results. So one of the first results they found correlates to figure 1, and this shows that the luminescence from the parasites post-infection and how it travels to the brain over time from the beginning of the infection. So the toxoplasma caused tissue cysts in several regions of the brain, but they were most prevalent in the amygdala, which is the emotion regulation center in all animals, and this could explain the lack of aversion to predatory olfactory cues. Figure 4 showed another result where the toxoplasma overall the infection did not affect any anxiety behaviors of the rats when in the open field area. So there's very little difference in difference traveled, time spent in the center, and the number of progression segments between the control rats and those infected with the parasite. So this shows that there was no real effect from the infection of the normal exploratory behaviors of rats and mice, which supports the behavioral manipulation theory. Figure 3 showed results that the infected rats spent overall more time than the control rats in the area with bobcat urine cues and less time where there were rabbit urine cues. So the aversion to bobcat cues became slight attraction, and there was a slight aversion to the rabbit cues that developed. So this shows that the toxoplasma did affect fear in rats that they have towards the predatory cat cues. And this could be inferred that by suppressing the aversion to cat odors, the rats might be more likely to come into contact with and be killed by predators. So while this decreases fitness in the rats, it increases fitness in the parasites, because overall they want to end up in the cat's stomach so that they can reproduce successfully.
More results to come. The aversion of cat odor was specific, and the other fear anxiety behavior stayed the same. There was no influence on the conditioning or the evolution of the fear of the infected rats. The infection did not cause any neophobia or hippocampal learning, and the infection did cause brain cysts in several areas, but were mostly focused in the amygdala. These additional results show that there was an aversion to the cat odors was changed, and the other fear conditioning was not affected. Conclusion: Toxoplasma affected in the wild rodents reduced aversion to the predatory odors and developing a mild attraction towards the bobcat urine, but did not compromise learned fear or anxiety in the subjects. Toxoplasma alters the behavior of the host for its benefits according to the behavioral manipulation theory. Overall, this experiment provided evidence that the amygdala, where most of the cysts were found, affected different emotions like anxiety and fear. Because of the only fear towards a predatory odor was affected, the parasites only wanted to affect the behaviors that were beneficiary to them. In this case, that would mean the parasite would be able to move up to a bigger host. Other general behaviors such as grooming, eating, or locomotion were not affected. These could be inferred that because the cysts were not there and they were only in the amygdala region, also because the behaviors were not tested to appear that there were no results of them. So for the broader context of this, there are approximate mechanisms. There are two general possibilities of why the cysts appear where they do. So does toxoplasma specifically select an area of the brain where it's going to be infected or where it's going to infect? Or is it just kind of a random site? As mentioned earlier, they affect mostly the amygdala, which is right down here in the temporal lobe, which is in the hippocampus, so it should be right in here is where the amygdala is. And that's, again, where the cysts will typically be found.
So continuing the broader context, there were two Tinbergen questions asked: behavioral causation and behavioral function. So what causes the abnormal behavior? So is it just the presence of parasites in the host body? For example, is the infection itself enough to change behavior because the nervous system is trying to attack the infection present? Or is it the presence of a brain cyst? And then does the specific area where the cyst locate matter, as we've talked about? So for behavioral function, what's the function of the behavior? And it's been hypothesized that with the Toxoplasma infection, there'll be an aversion to the feline odors, and that becomes an attraction. So that's what it does to the hosts. So this overall experiment provides support for the behavior manipulation theory. It only changed the aversion to predatory odors and not the learned fear and anxiety in the subject rodents. So the infection did not influence fear conditioning or its extension in rats. So comparing control rat to infected rat, there was no significance between the two. Other conditioned fear and anxiety, such as neophobia towards new food, was not affected.
Continuation of this theory, it's only been known that the parasites can affect the host behavior since about the 20th century. As I said earlier, it is, new and prevalent now. So the why and how of all of this is still really isn't understood that well, and rats can get affected with the parasite, will cause them to be attraction of the odor of the bobcat or the cat, as mentioned. It also increases the dopamine in the rats. This we'll get to a little later. Humans can get the parasite by eating undercooked pig and lamb, as mentioned earlier, and also unwashed produce. This then can lead to Parkinson's and psychiatric disorders like schizophrenia and vegetative disorders in humans as well. So for future research, as mentioned earlier, it can affect humans. So when Toxoplasma has a causal relationship with schizophrenia in humans, and not much else is known about this, but there's a correlation there. And it was interesting, with a low amount of Toxoplasma in a human host itself, it can be a high association of dopamine in the midbrain, which is interesting because, as earlier said, in rats, they have an overall increased high dopamine level. So, can non-humans develop schizophrenic type behaviors? So, I guess the operant definition of this would be: can the animals be able to get this? And we want to identify what is this type behavior. Also, is attaching something like this anthropomorphizing the animal, or is it just taking it somewhere else? Another thing for future research could be how toxoplasma affects other hosts and how their behaviors as well. So another thing is, do they have a preference in the host? Is it rats versus mice or humans versus pig? Stuff like that. If so, what is the reason behind it? Does it have anything to do with the brain composition of the host, or does it have to do with anything, maybe even the size of the host and the damage that they could do? So continuing more with humans on this, human hosts can have stillborns if they're infected so much with the toxoplasma. We want to know, does this also happen with the other hosts? So if you have a pregnant female rat, for example, would that affect, the pups in the litter of that one? Possibly same thing with the mice. With the human host behavior, infected men have shown to have behaviors of jealousy, more introspective, slower reaction time, lower self-esteem, disregard social rules, and infected females have shown to have higher behaviors of higher self-esteem, loyalty, respect for social rules, and effectiveness. So it's curious now, does this also happen in other rats and mice when they get affected as well?
Finishing up future research, there's a couple more questions that we were interested in. So why do the parasites cause cysts mostly in the amygdala? Does it have anything to do with its control or impression on the emotions of a host? The payoff between beneficial behavior for a parasite and the undesired behavior of the host. Is it possible that the increase in predation is worth it for the parasite? So, for example, what if the parasite doesn't get a chance to reproduce because it was predated on, or the predation occurred with a different, unwanted predator? And are there any evolutionary adaptations to prevent the infection of toxoplasma in rodents? So could the host's brain develop adaptations that would attack the infection and or the cysts and kind of kill it before it really caused any of the behavior manipulation? Does the cyst density affect how severely the behaviors change? For example, would a larger cyst in the amygdala region cause less aversion to cat odors than a smaller amygdala cyst? And a study examining this could begin to look at how sensitive the brain is to biological changes.
For our references.
Thanks.
Have a good one.